Okay, good afternoon. Welcome, everybody. It's been a long season of webinars, and this is the penultimate session we are having, and we are going to have a fireworks session because today we have the privilege to host Professor Michael Anderson. Let me briefly introduce Michael Anderson, although he doesn't need any introduction. He is known worldwide. He is professor in philosophy of science at the Department of Philosophy of Western University, London, Ontario. And his research is located at the intersection of psychology, neuroscience, computer science, and the philosophy of cognitive science. His most recent book, which I warmly recommend to all of you, After Phrenology, Neural Reuse and the Interactive Brain, was published by MIT Press in 2014. And this book outlines a novel framework for understanding the overall functional organization of the brain, places its function in evolutionary context, and demonstrates how mechanisms originally evolved for the support of sensory motor coordination have been co-opted to facilitate language and higher cognitive functions. Michael was, in 2012-13, a fellow at the Center for Advanced Study in the Behavioral Sciences at Stanford University. The title of his talk today is Neural Reuse, Dynamics and Constraints, Getting Beyond Componential Mechanistic Explanation of Neural Function. We are all ears, and Michael, please, the screen is yours.
Okay, thank you so much, Vittorio, and thank you for the introduction. By the way, I can't see myself, so if something goes wonky as I'm talking, please let me know. I'm new to Teams. I've been on, as a lot of us, I've been on.
No, everything is okay. We can see both you and the screen.
Okay, I've been on Zoom for a very, very long time, but this, I think, is my first foray with Microsoft Teams. In any case, also, I think we have a relatively small crowd today, so please feel free to interrupt me, stop me to ask questions and whatnot as we go. This doesn't have to be a kind of a big formal lecture. As Vittorio mentioned, a recent book, After Phrenology. The paperback actually just came out a couple months ago, so you can get it for less money than you could before, if you're interested. So a lot of the stuff that I'll go over here is covered in great detail there, and also a couple of these papers that I'm showing you right now that go into more detail on some of the subjects that I'll be talking about. So here's a basic outline of the talk that I'm going to offer to you today, just some basic brain facts, and then what componential explanation in neuroscience looks like. And then I'm going to talk about starburst amacrine cells as a possible counterexample that don't seem to fit very well into the componential mechanistic explanation framework. And then talk to you about what I call enabling constraints as a possible alternative or a supplement to componential mechanistic explanation. And then I'll finish with some hand waving about synergies and functional configuration space. Okay, excuse me, some basic brain facts. Everyone will be familiar with Hebbian plasticity. The slogan that gets used is that neurons that fire together wire together. In other words, that, you know, during the course of learning, some connections between neurons will be strengthened, other connections between neurons will be weakened. That's obviously absolutely crucial part of brain wiring and a crucial part of learning. What I've been pushing for is an alternate kind of neuroplasticity, which I've been calling neural reuse. And what neural reuse does is it acts on a higher level of organization and establishes multiple overlapping neural coalitions for different purposes. So this figure here, find my pointer. This figure here, which maybe you'll see a few times in this talk, you can just imagine a very simple brain with these 6 regions defined however you like. And the regions cooperate with one another under different circumstances. So the outline here, 1, 2, 3, 4, 5, 6 are on the outside. Maybe that's some kind of sensory function. And those brain regions talk to each other in order to achieve a particular kind of perceptual capacity. But those same regions, or many of those same regions, will also be involved in, say, the motor response to that perceptual activity. And that's sort of represented here with the red. So the patterns of cooperation will differ, although the same brain regions will be used over and over again across multiple different circumstances. So that's neural reuse. It's a kind of plasticity in the brain, and I think, again, over the last decade or so, I've been establishing that it is a crucial part of learning. And neural reuse then leads to a very specific neural architecture, whereby the mapping between cognitive processes and brain regions is many to many, whereby individual regions of the brain are used and reused in diverse tasks across task boundaries. And differential function then is not so much the result of differential activation of individual brain regions, which has been the classic picture, but instead differential function is the result of dynamically combining the same elements, but in different coalitions. So I'm going to talk a little bit about how we establish the evidence for this sort of claim. And that's called a functional diversity analysis. And the steps here are relatively straightforward. You build a large collection, or rather you have your grad students build a large collection of brain imaging experiments. Right now my database has around 6,000 experiments in it. Then you categorize those cognitive processes, and you can do this in various ways. So you can imagine, motion tasks, or you might do more fine-grained than that, fear and love and whatnot. A visual perception, auditory perception, however. You'll see as I go that we've done these kinds of analyses with different levels of grain. The qualitative results come out the same every time we do it. Anyway, then you calculate the diversity of function observed in each part of the brain. What does that mean? Well, we borrowed a measure from ecology. Ecology is a field that obviously cares a bit about, say, species diversity in a particular bit of the ecosystem. We just applied this way of measuring diversity to the brain. So the analogy is quite straightforward. If you're an ecologist and you're looking at a particular bit of the world, if you see many plants and many animals from many different species, that's going to be a high diversity area, the rainforest, for instance. If instead you see that most plants and animals are in a single or just a very few species, maybe in the desert, for instance, that's going to be a low diversity area. Now, we've scaled this measure, the Shannon diversity measure, from 0 to 1 to make it sort of easier to interpret. So something that's low diversity is going to be closer to 0, something high diversity is going to be closer to 1. Again, just to make the analogy explicit, if for a particular region of the brain we see many cognitive functions from many different categories, that's going to be a high diversity region. If instead, in a particular region of the brain, we see most cognitive functions of being in a single or a few categories, that's going to be a low diversity region. This is, I'm afraid, I'm not quite sure who the audience is, but I always like to be very clear about how to read the data that I'm showing you. And I'm just going to show you the histogram of what we did. But I just want to make sure everyone is on board with what that's going to look like. So this is a histogram. This is how to read a histogram. And a histogram is simply a way of representing the values that you're measuring compared with or represented as the number of times you see a particular value. So suppose that I am measuring the heights of people in this virtual room. Well, so the first person I measure is on the, say, shorter side of things, and so that's where they land, and the next person I measure is sort of an average height, and that's where they land, and so on. We get a person on the shorter end of things, and so on. Anyway, the idea is that you keep measuring whatever it is you're measuring, and that gives you a distribution. And that distribution tells you something important about the underlying phenomenon. So here's a kind of distribution that people will be familiar with. These are the SAT scores. This is I think it's not much used in Europe, but it's crucial to the United States college education, college admission system. And so this gives you a histogram of SAT scores. Now, this is a nice normal distribution, and it's a normal distribution because, of course, they control to make sure it's a normal distribution. But the idea is that the typical person, the average person, scores around 1,500 on this score. And if you're really talented at this particular kind of test, you might score upwards of closer to 2,400, and if you're having trouble, you might score down on the low end towards 600. Here's another normal distribution, which is sort of interesting. Someone decided to ask people on the street, how much money does Brad Pitt carry in his wallet? And that's a nice normal distribution centered around about $500. This is ridiculous, of course, because Brad Pitt doesn't carry any money, I'm quite certain. But nevertheless, this is what people say when you ask them that question. More important are going to be distributions like this that have a skew to them. This distribution, and ignore the last two bars on the right there, the bin size changes when we get that far out. But here's the distribution of household income in the United States. And you can see it's highly skewed, and most of the values are on the lower end because income in the US is very unequal, as you will probably be aware. And so the idea here is that tells you something important about the US economy, who it's serving and who it isn't serving. And you can see this just from the histogram. This is the reverse skew. This is test scores from a recent test I gave in cognitive psychology. This was at Franklin and Marshall College, an elite liberal arts college in the United States. And I have good students. So in contrast to the income, they instead are clustered towards the high end of things. And that's the point I want to make here. So a brain with mostly specialized regions will look more like the distribution of household income in the US. And a brain with functionally diverse regions will look more like this. So the picture, the histogram I'm about to show you, that particular one was produced by taking every voxel in the brain. So as some of you will be aware, a voxel is a volumetric pixel, and it's the basic unit of measurement for fMRI studies. So you look at each part of the brain, each location in the brain. So I took each voxel in the brain, and I drew a sphere around it, in this particular case, of 10 millimeters. And then I just looked, well, what activated that sphere of brain tissue? And as I showed you before with the Shannon information, I calculated the diversity of activations within that region. I did that again for every single location in the brain. And I think in this particular case, I'll remind, I think I did 12 different categories. Again, I've done this with different sizes of spheres. I've done this with different numbers of categories. Qualitatively, the results come out the same, which is basically this. You see this massive skew towards the upper end of the scale. In other words, the vast majority of the brain is highly diverse. It's serving, each part of the brain is serving multiple different cognitive functions in multiple different categories. To give you some way to interpret this more specifically, I'm showing you these spider plots. So a spider plot, right, is just, so again, we have 11 categories in this particular run, and the names of the categories are here. 11 categories in this particular run, and the distance out from the center tells you the proportion of activations in each of those categories. So this one here is a voxel from left anterior insula, and it has a diversity value of 0.88. Now that's, as you can see from its location on the histogram, that's a relatively unusual and relatively high diversity. But the point here is that it's basically activated by every category except for vision, right? That's 11 here. So it's almost equally activated by all these different categories. In contrast, here's a voxel from left auditory cortex. And this one it has a 0.41 diversity because it has a little bit of activation in a couple of things, but it's pretty much dedicated to audition, right? So that's a relative specialist in the brain. Now, importantly, you'll see where 0.41 falls on this histogram. So although there are relative specialists in the brain, almost none of the brain looks like this. And this is a surprise to many these sort of classically trained neuroscientists, where we expect kind of functional dedication. This is not what we see when we look at thousands and thousands and thousands of neuroimaging experiments all at once. Just to round it out, this is a voxel from left thalamus. It has a diversity that's basically the median for this skewed distribution. So this is what a typical brain region looks like. And you can see why it sort of prefers somesthesis, bodily perception. It's also activated across a number of other categories. And that's what a typical brain region looks like. Each brain region is typically activated by multiple different kinds of cognitive functions across multiple different categories. This is the spider plots from a different run. These happen to be 6 subdivisions of insula. Doesn't much matter what exactly they are. So this is, I think, 22 different categories, so a bit more fine-grained than the one I showed you before. But here the idea is just, I want to make a simple point, which is that we definitely have functional differentiation in the brain. Nobody's going to claim that each part of the brain does the same thing in the same way, right, all the time. That's not true. We have functional differentiation, absolutely. Each part, right, of the insula here looks very different. It has a very different mix of cognitive capacities, cognitive functions that it supports. Nevertheless, we have typically, again, classically, inferred functional specialization from our observation of functional differentiation. And that turns out to be an unsupported inference. We absolutely have functional differentiation. Each bit of the brain is different from each other bit of the brain, but very little of the brain is highly specialized. Okay, so that's the functional diversity analysis that establishes.
Sorry, Mike, can I ask a question?
Absolutely.
I was wondering whether the result would stay the same if you do the same analysis with, instead of MRI activation, for example, the behavioral results, the behavioral outcome of brain stimulation with, for example, intracortical stimulation, surface stimulation, so some kind of causal approach that reveal the function through a perturbation on the behavior. Would it stay the same? What do you expect?
I do expect it would stay the same. So I have not done that kind of work. Let me give you the reverse thing. It's not a causal thing, but even if you do this study for individual neurons, and I think I have some results on this later in the talk. So for instance, canonically, hippocampal place cells, right, which are supposed to activate just when an animal is at a particular location in its environment. Well, it turns out hippocampal place cells are also activated by odors. They're activated when an animal is at the location, before they're at the location, after they're at the location, and then the other, again, more causal now, brain injuries, traumatic brain injuries. Again, canonically, people talk as if a brain injury will knock out one very particular function, but of course, anyone who has worked with patients knows that's not true. Any individual, there's some deficits from an injury might be more salient, might be more important than others, but basically any focal injury is going to affect multiple different kinds of cognitive and behavioral functions. So I would think TMS or whichever sort of knockout technology you favor is going to give you the same thing. And you can, a lot of TMS experiments focus on, oh, well, I'm going to knock out this so they don't notice a particular stimulus. But if you measured, if you change your dependent variable, the claim is, and I think there's good reason to expect this, the claim is that you would discover multiple deficits from knocking out any particular region of the brain by whatever technology you favor. Does that work?
Yeah. Okay. Thank you.
Yeah. Good. Thanks for the question. So anyway, so what I showed you now is the evidence up to now is the evidence for functional diversity, but we still have to explain how differential function comes about if it's not a matter of differential activation of individual regions, again, as the classic neuroscience picture tells us. So instead, we say, well, it's the reason you get differential behavioral outputs, differential cognitive results, is because the same regions of the brain, these functionally diverse regions, combine in different coalitions to produce differential function. So how do we know that? Well, so to figure that out, what we do is we look for evidence of statistical dependence in the activity of different brain regions. That is, does activity in region A make activity in B more likely, and the reverse, and so on. For those interested in the statistics behind this, we basically are doing a chi-squared analysis and looking for deviations from randomness in the activity, in the mutual activity of brain regions across different conditions. And if we see this deviation from randomness, then we say that those regions are functionally connected, that is, they act as partners for a given task. And the results of those kinds of analysis can be represented as a graph. Now, mathematically, a graph is an abstract structure combining a set of nodes joined by edges, where the nodes represent usually objects and the edges represent relationships. So I'll give some examples here in a second, but examples of graphs include airline route maps, social networks, anatomical connectivity maps, and we're going to show you again, the whole point here is to show you a functional connectivity map and look at some of the interesting patterns that we see there. So look, here is an airline route map. Well, it's a fragment of an airline route map. Those of you who have flown through the United States will recognize this is likely the Delta route map centered on Atlanta, which is where their hub is. And in an airline route map, the nodes represent airports or cities, and the edges represent the availability of flights between them. Here's a fragment of a social network, Facebook or whatnot. And in a social network like this, the nodes represent individuals' accounts or people, and the edges represent whatever relationship they have, likes or friendship or whatnot. Here is an anatomical connectivity map where the nodes are brain regions, and the edges represent, in this particular case, the existence of white matter connectivity between them. And by the way, there's more information here then we're going to discuss. The thickness of the lines indicate the degree of connectivity, how much white matter connectivity there is. So I'm going to be focusing on really simple relationships, just because it makes the outcome clearer. But graphs are really quite a useful tool for representing patterns of relationships between entities. So here are the nodes that I'm going to be using. Let me orient you to this. We're looking at the brain from above. It'll emerge as you look at this for a bit. The shape will emerge. We're looking at the brain from above. This is left and right frontal pole, right? So at the front of your brain, this is left and right occipital pole, so the back of your brain. And each of these spheres is organized, is placed at the anatomical center of the, I think, 113 regions derived from the Harvard-Oxford Brain Atlas, Anatomical Brain Atlas. So, whoops. And so what we're going to do now is we're going to project onto these nodes, each of these nodes being a particular region of the brain according to the Harvard-Oxford Atlas, and we're going to project the relationships under various circumstances. So this is the relationships, the functional relationships that you observe during semantics tasks, right? So again, we're looking for deviations from statistical randomness, which regions tend to be active more often according to the chi-square analysis, and this is under semantics tasks. So that is tasks involving language meaning. Now you can only probably sort of see the massive amount of connectivity that you see across the entire thing. I don't know how good your projector is there. But what I want to do, however, and let me just say one word about that. So we tend to think of language as being a left lateralized function in the brain. And again, I'm not sure if you can see this very well given how this is projecting, but indeed you see some evidence for that in this. There's a very high density of interconnectivity on the left side of the brain. But there's also massive amounts of colossal activity. A lot of the brain is involved in language tasks. This shouldn't be surprising, right? The notion that we have a singular or a few regions specialized for language, given how complex language is and given how important it is to us as a species, was probably not the best hypothesis to begin with. In any case, so you do see evidence for this left lateralization, but you also see a much more complicated picture that emerges when you look at what brain regions talk to which other brain regions during tasks involving language meaning. Now, for the purposes that I have here today, I have highlighted the partnerships of left precentral gyrus. There's a whole nother story to tell about why left precentral gyrus, but for us it doesn't quite matter right now. I just want you to notice that left precentral gyrus, right, has a very particular set of partners during tasks involving language meaning. Now I'm going to switch the picture to tasks involving emotion. Again, I'm not sure how well you can see this in detail, but we do talk about emotion as being subcortical, importantly subcortical, and indeed you see evidence for that in this picture. There's a high density of interconnectivity between subcortical regions right there in the center of this sort of abstract brain. But here again, but as you can also see, I hope, just massive amounts of connectivity across the entire brain when we're looking at emotion tasks. And again, this shouldn't be surprising, again, given the centrality of emotion to our to human experience. Nevertheless, again, back to the simple thing, here I'm emphasizing left precentral gyrus. And you see that it has its particular set of partners. I'm going to go back up to language for a second. And I just want you to notice that, yeah, left precentral gyrus is involved in both emotion and language tasks, but it has a different set of partners in each condition. Now I'm going to go for the third and last example, and this is stimulus-driven attention. And again, left precentral gyrus being highlighted here, right, has its set of partners. So left precentral gyrus is involved. In fact, many of these nodes are involved in all of these different functions, but the partnerships it has are different in each. I'm going to just run through those again. And here again, qualitatively, just the idea is you can see that the fan of connections changes as you change the condition under which you're viewing the functional connectivity in the brain. Now, we don't have to stick to just the qualitative measure, right? We can actually give you a metric for this kind of neuromodulation. And one point I do want to make, although I won't emphasize it very much here, is that this large-scale neuromodulation, which is sort of characteristic of neural reuse, as I formulated it, there's actually compelling evidence for neuromodulation at all spatial scales. So between individual neurons, between regions, even in terms of genetic expression, right? So this notion that there's reuse of individual, say, biological elements seems to be a feature of biology across different spatial scales. Okay, but as here I forgot I had this slide in here. So here's an example. I'll just mention it. So this is each of these triangles represent a neuron in C. elegans. C. elegans being that soil nematode for which we have a complete map of its neuron, right? Of all of its neurons and all of its connections. So let's just focus on ASH here. This is this red one. So ASH is a single neuron that participates in multiple different behaviors. Octanol avoidance. This is a noxious substance in the environment that these nematodes don't tolerate. Aerotaxis, that is looking for air. They do require oxygen to survive. And then a particular set of social behaviors, aggression and repulsion. So ASH, for instance, is involved in all of these behaviors. However, as is shown in these diagrams, it participates with different partners in each of these, and which partners it participates with depend on neuromodulatory effects, and in particular genetic expression. So NPR1 being expressed or inhibited and so on will change which partners ASH has under these different circumstances. And again, there's a lot more to be said about this. I go into great detail on this sort of thing in the book. But here again, the point is just that this notion of reuse seems to be an ubiquitous phenomenon across different spatial scales, which makes total sense in the sense that, you know, neural tissue is expensive. Neural tissue, it only represents a small fraction of what's available to us resource-wise in the body, and it has to do a lot of really complicated things. And so the notion that you would not dedicate individual bits of it to individual outcomes, but instead would use and reuse each individual element across multiple circumstances makes sort of better makes better sense of efficient use of a scarce resource. I'm going to ignore that. Okay, so that's the kind of background of basic brain facts. Now I want to talk briefly about a particular style of explanation in neuroscience that I think neural reuse puts pressure on and asks us to move a bit beyond. So here's a summary so far. Individual regions of the brain have multiple high-level uses. The brain achieves its function by sticking together the same parts in different configurations to get the job done. And there is both significant functional differentiation and significant functional integration, but there is not significant functional specialization. Now, these facts and others that I'll mention as we go, I think put significant pressure on the leading account of explanation in neuroscience, which is componential mechanistic explanation. And I'm thinking in particular of people like Bill Bechtel and Carl Craver. So here's a quote from Craver. He says, componential mechanism construes explanation as a matter of decomposing systems into their parts and showing how those parts are organized together in such a way as to exhibit the phenomena of interest. Scientists discover lower levels by decomposing the behavior of a mechanism into the behaviors of its component parts and decomposing the behaviors of its parts into the behaviors of and blah, 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 right? You see how this is supposed to go. So this is a kind of a decomposition and attribution story. And I think already you can see that that gets a little bit problematic, given that as you decompose, it depends on what circumstances you're looking at a particular part as to how you're going to describe what its behaviors are. Now, and this is an important sort of a part of their view, that function emerges from interacting structure in a bottom-up fashion. In other words, once you've gotten to what the behavior of a particular region is, then you just add that all up together, and that gives you the behaviors of more complex organizational structures. And again, I think that what we're seeing is that one will not, in fact, cover all the cases that we can observe of how brains work. So this is just the Craver's summary. He says, look, here's what you do when you want to explain things mechanistically. You identify the phenomenon of interest. Psi, you define the system S that psi's. You identify the relevant spatial subparts. I call this set X of the mechanism and the relevant properties and behaviors. And then you describe how the parts are organized such that the system exhibits the explanatum phenomenon. Carl's well worked out example is the action potential, right? So, okay, the phenomenon we care about is the action potential. The system that exhibits that is the neuron, and so on and so on. Now, what I want to say is that the ubiquity of functional diversity and the ubiquity of neuromodulation puts pressure on this account of how function arises from interacting structure. And importantly, by the way, and again, I'm not quite sure who the audience is here today, this is not just a philosophical point, right? This is going to change how neuroscientists interpret their own data. Because if they observe behavior, that doesn't mean that's the behavior, as I'll emphasize later in this talk. That doesn't mean that's the behavior of the system that's going to exhibit under all circumstances. If we have ubiquitous neuromodulation, then any given particular bit of the brain that any neuroscientist is interested in studying is likely going to have different behaviors under different circumstances. That makes science quite a bit more complicated, unfortunately. But if we want to get at the ground truth, that's just what we have to deal with. And so here, that's exactly the problem, as I say here on this slide, that The problem here is that it seems to assume that we have identifiable subsystems with fixed behaviors. That is, we can come up with simple attributions of function to particular identified subparts of the neural system, and that does not appear to be true. So we ask, okay, what's the behavior of left dorsal anterior insula? Well, here's dorsal anterior insula. It's active across, a dozen different circumstances. So what's the behavior? I don't know. It's a behavior under a circumstance, in a circumstance, and we have to understand that those contexts are crucial to understanding brain function. What's the behavior of neuron ASH and C. elegans? Well, gosh, I don't know, right? It's involved in multiple behaviors. And we just need to take account of that when we're trying to explain how complex behavioral and cognitive function arises from interacting structure. Now, again, assuming there are philosophers in the audience here, definitely we can save the picture of mechanistic explanation by weakening the requirement for mechanistic explanation to say, oh, well, I just need to identify the behavior given at that is fixed at a given time or in a given context, and that's true. But what I want to suggest is maybe it's time to rethink the issue in a more systematic way. And one motivation is because that begs the question of how these component functions get temporarily fixed in the first place. From the standpoint of explanation, you could say, yeah, okay, I'm only going to explain function under the circumstance that I'm particularly studying, but you still would like to know, okay, that's fine. How is it that that particular bit of the brain had that function at that time? How did that happen? So here's a particular example. This is again, a spider plot. You're used to seeing these by now, of the dorsal attention network. And again, the important thing is that the dorsal attention network is, at least by hypothesis, dedicated to attention, but it's not composed of attention-specific parts. As you can see from this spider plot, the nodes that make up the dorsal attention network have functions that range across multiple different circumstances. But let's just assume the hypothesis is true, that the dorsal attention network is, as organized, dedicated to attention. Well, you have to ask, how the heck did that happen? And so there's more kind of examples like this. I'm going to ignore this because I've been going on for a while here. Let me just skip by this. I mentioned this already, so I will say a word about this. This is the activity of hippocampal place cells, again, canonically active when an animal is at a particular location in its environment, and that's true. These cells are active when an animal is at a particular location in the environment, and that's exhibited by B here. But it turns out these things fire also before you get to that location in the environment, and they also fire after you leave the location in the environment, with the difference being on the relationship, temporal relationship in that firing to the background oscillation of the hippocampus itself. Right, so we got sort of radical context dependence of function. What it is for a place cell to be firing depends not just on the environmental circumstance of the animal, but also depends on the neural, as it were, circumstance. In this particular case, it depends on the oscillations, the background oscillations of hippocampal cell firing. The point is, all this puts pressure on the notion of components. Right? If the output of or the behavior of a part of the mechanism differs based on the background conditions, whether environmental or neural, it's not clear anymore what the claim to have identified a component function comes down to. And that raises the possibility, the important possibility, that there may be a violation of the bottom-up assumption of componential mechanism. That is, where you can identify the particular behavior of a particular part and then just sort of add up those things and show how they give rise to higher-level function. And this is where I want to go quickly then over Starburst amacrine cells as a kind of case study of a possible counterexample. to the mechanistic explanation, the style of mechanistic explanation here. So here is a picture of a starburst amacrine cell. You can see why they're called that. These are really super interesting cells. So these are cells in mammalian retina. They've got we've got the soma, and we've got a bunch of different dendrites. Now, interestingly enough, there are no axons in a starburst amacrine cell. The dendrites act sort of both as dendrites and as axons. That is, they accept neurotransmitter and transmit impulses to the soma, but also release neurotransmitter to neighboring cells. So they're kind of a interesting, bizarre hybrid cell, and of course they're called starburst amacrine cells just because of the anatomy you can observe here. So they're super important to motion perception, and the way they're important to motion perception is each starburst amacrine cell dendrite is a directionally selective spatial subpart of the overall cell. That is, each dendrite tends to release neurotransmitter in response to motion that occurs in its preferred direction, that is, from the cell body outward. Now, importantly for the story I'm going to tell you here, the SEC network in mammalian retina is quite dense. We have lots and lots of cells in a densely overlapping network, as is shown in this picture here. And again, to make a longer story short, the way this works is that each of these cells both excites and inhibits its neighboring cells. And that's important. So the idea here is that as you get, say, light moving across the retina, this starburst amacrine cell, right, which is responding to the light moving across in its preferred direction, will actually inhibit this starburst amacrine cell. So this cell will respond to the light moving in its preferred direction, whereas this dendrite will be inhibited by the neighboring cell. Why is this important? Well, again, in the story I'm telling, this is important because if you take a starburst amacrine cell in isolation and you, say, stimulate it with light moving in a particular direction, all the dendrites will respond to it. That is, in isolation, these cells are not, in fact, selective. The dendrites are not, in fact, selective. They are only selective in the context of the network. And what that suggests to me is that we cannot tell a componential mechanistic story about these cells because the components of these cells are not functionally selective. The components of these cells are only functionally selective in the context of the network in which they exist, which is a top-down effect, not a bottom-up one. So whereas it would be natural to explain direction selectivity in terms of the intrinsic or lower-level properties of the dendrite, this kind of functional attribution, that doesn't look to be actually true of these. That's not a good explanation of how these things work. In fact, an important part of the mechanism is extrinsic and at a higher organizational level than the component parts themselves. Again, this is a longer story I'm going to skip about direction selective ganglion cells, but the same kind of story applies there. The point is that these examples warrant an amendment, at least, to mechanistic explanation, because the parts of the mechanisms in virtue of which systems exhibit behaviors are not spatial subparts of or lower-level components of the system. That is, sometimes they operate with the imposition of functional constraints coming from whatever spatial level. In this particular case, there are functional constraints imposed by the organization of the overall network. Okay. Where I want to just introduce the term enabling constraints as a supplement or maybe alternative to componential mechanistic explanation. And again, just reminding you, right, we've got these individual neurons that are active across multiple circumstances, but do different things in different circumstances. So ASH, for instance, has the same capacities in all its networks. It's a cell. It does whatever that cell is capable of doing. But these capacities, although they're necessary conditions for functions, they aren't yet functions. They aren't functions in the relevant sense that lead to, say, behaviors, to psychological outcomes. Just as direction selective ganglion cells in the absence of starvus amacrine cells will continue to exercise their capacity to fire in response to light, but they're non-functional because they're not actually telling you anything. The idea here, again, is that constraints in the brain limit capacities so as to enable function. And this is a different kind of explanation from componential mechanistic explanation. Whereas in componential mechanistic explanation, you identify function, and then you build up function into more complex things. What I'm suggesting here is that no, actually, there's also a role for constraints between different parts of the brain, and those constraints create or enable function, right? It's not that these parts of the brain have function natively. Those functions are enabled by their relationships with other parts of the brain. And this is just the formalization of this, which I'm going to skip a little bit. I'm just going to skip the, if people want me to go through the kind of philosophical formalization of that idea, I'm happy to do that in the question period, but I think the idea is clear enough for our purposes here. Now, I think enabling constraints as explanations have some advantages over mechanistic explanations. First of all, they're synchronic. That is, all mechanistic explanations are diachronic in the sense because they're causal. Right, this has to happen, then this happens. Whereas enabling constraints are synchronic. That right, individual bits of the brain can mutually constrain one another at the very same time. There doesn't need to be a kind of a temporal order to that constraint. And again, for that same reason, they're potentially symmetric, right? You can have mutual constraint between different parts of the brain. And constraints easily accommodate various kinds of interlevel explanations. Those who are familiar with this literature will know that the mechanistic explanation folks have big trouble with top-down explanations and try to get rid of it in various ways or accommodate it in various ways. Constraints don't have this issue at all. And of course, constraint applies at multiple spatial scales. So back to this dorsal attention network, the idea is that maybe the notion of constraint can help us explain the emergence of selectivity in large-scale networks with non-selective parts. The idea is, of course, that each individual node in this selective network mutually constrains the other nodes, and that's why you get selective function out of functionally diverse, functionally differentiated parts. Right, so the parts of the network interact so as to impose these mutual enabling constraints. The way we can think about this is this might act to selectively inhibit and enhance some subset of the existing capacities, and that leads to networks with specific functions. And I'm going to end on this just to bring to the table a very old notion, which is that of a synergy. And this classically comes to us from motor control, but it applies more broadly. And a synergy is a functional grouping of individual elements, be they neurons, muscles, limbs, individuals, whatever, that are temporarily constrained to act as a single coherent unit. And in the brain, there's mounting evidence that one effect of task-related stimuli is indeed a reduction in the variability of neural activity, which is characteristic of the existence of a synergy. So Bernstein, in his classic motor control studies, showed that if you're hammering, and say you're an experienced blacksmith, and you're hammering at a particular object, the variability in each of the joints is actually greater than the variability of the strike location, which is kind of interesting, right? What that means is that the variability in each of the individual joints in this motion, each is compensating for the other variability so that the hammer strike is consistent across time. The idea is that the same is true in the brain, that when the brain is focused, when a person is focused on achieving a particular task, individual parts of the brain mutually constrain one another so as to enable the consistency of behavior that is necessary for successful responses to the environment. Again, some details that I'll skip. Again, so this is just summing up real quick here. Many explanations in neuroscience attempt to identify components and their functions, but I think that empirical results are putting pressure on the adequacy or generality of that approach. And that includes the reuse of local regions in multiple situations, dynamic neuromodulation, interlevel interactions, context-dependent functional attributions, nonlinear interactions between evoked and intrinsic activity, which I didn't go over in detail, but have some work on that. And here then is the kind of methodological point. These emerging approaches, this emerging evidence, I think, is more consistent with dynamic systems theory than with more linear models of brain function. And so the goal here should be less about functional mapping. Let's decide which bit of the brain does which thing, and more about understanding the parameters and constraints that govern the behavior of the system. Or again, it's just highly abstract, but the idea is that the neural system, traces a trajectory through a high-dimensional functional space, and what we want to understand are the sets of constraints that shape that trajectory. And again, for more details, here are a couple of options. As Vittorio mentioned, the book After Phonology, as I say, the paperback just came out a few months ago. And these two articles, there's more. If you just go to my Google Scholar page, you can see even more recent stuff than this. And that brings me to the close. Thank you so much for your attention.
Well, thank you so much for this exciting and provocative talk. Just before our audience warms up, let me ask you a few questions. So, from what you said, and also from what we see by reading the neuroscientific literature on a daily basis, it seems that we are still with both feet in a sort of high-tech version of phrenology.
Yeah.
So the first question is, how is that? What is your explanation of the fact that so many colleagues of ours are still willing to support the notion that phonology is there, syntax is there, semantics is over there, the mind-reading lobule is there. Also, I gather that even if we move at lower scale of investigation, none of us is in principle immune to the limitation that stems from knowingly or unknowingly somehow obeying to a mechanistic level of explanation. But so just to start, what's your book came out in 7 years ago, but there's still much work to do, apparently.
No, absolutely. Look, the initial response to the work that I did, this is back before the book, was mostly dismissive. And then as the evidence mounted, it's been mostly acceptant, but as my friend Lucino Udin says, it's kind of acceptant resistant. Like, yes, well, we see the evidence, and so yes, indeed, bits of the brain must be multifunctional in the way you're describing. But nevertheless, I'm really not going to think too hard about what the implications of that are for scientific practice. And why is that? Well, I mean, the simple explanation is people are busy and they have their specialties and they're doing their work. The more complex explanation is, and this is why I put in the very end of the book, the very last chapter, is all about what we can do instead. Right, here are the alternatives. Here's the alternative methods we can use. Here are the alternative questions we can be answering or asking. Because I'm well aware that if you just criticize somebody and don't give them a different thing to do, nothing is going to change. And it's an unfortunate fact that I think the positive part of what I've been putting forward, the reuse and diversity studies and the functional connectivity and whatnot. That's been taken up, in quite broadly. But unfortunately, the methodological part has not had the impact that it would, I think is important for the field. So, I myself, we've been a bit stymied by COVID, as I'm sure is true in Italy as well. I've had some experimental projects have to be put on hold for some time. We'll hopefully get back to them this summer as we're able to do in-person experimental procedures. But, so one of the things I'm trying to do is not just say, hey, we could do this, but actually do the thing. And so we've got a really nice project, I think a really nice project involving a combination of virtual reality, behavior, and EEG, where what we're looking for is, this isn't a super simple behavioral task, a FITS task if you're familiar with it, but it's basically just a rhythmic sort of pointing thing for which the behavioral dynamics are quite well understood. And then we can control, with VR, we can control the perceptual information that's involved in enabling that particular task in a quite precise way. And then with EEG, we can measure the functional constraints that the brain exhibits during this task. And so the idea is if we're successful, this will be the first experiment, to my knowledge, to bring all three of those things together: behavioral dynamics, perceptual information, and neural constraints in one picture, in one kind of explanation. And so if we're successful, that's a kind of a model, I think, for the kinds of things we can do in neuroscience that are sort of that better answer to the underlying empirical stuff that most people agree is real, but nevertheless isn't reflected in typical contemporary methodologies. So we'll see how that goes, and we'll see what the reception of that sort of approach turns out to be. Again, hopefully within the next year, we'll be able to get that out and into public hands. But yeah, I just think that's the kind of thing that we need to do. And just one more thing, because part of the limitation we're dealing with, look, people have been treating the brain, reverse engineering the brain, as if it's a 19th century engineering project, right? We know how to build trains, right, out of components, and we understand what the components do. And that's a very comforting epistemic position to be in, right? That's where the whole mechanistic explanation gets its traction, because you say, let's just decompose it into parts, figure out what the parts do, and then we can build it back up into something more complex. That's classic 19th century engineering mindset. We need to get beyond that, but it's hard to get beyond that because it's the fact is that's an incredibly useful epistemic mindset. It works in many, many different circumstances. It doesn't work for the brain, I don't think.
I mean, just to play the devil advocate, this epistemic approach to the empirical investigation of the correlation between brain-body states and behavior, however, provided us with results that, in a way, accomplished something in terms of, for example, diagnosis, take the direct indirect pathway to explain the physiopathology of Parkinson's disease. Yeah. I mean, now we are placing electrodes stimulating a deep structure that, although not all of the times, because of course people always tell successful stories, there are problems. But still, the treatment of Parkinson patient got improved from the knowledge about the functionality of the corticostriatal network. Yes. So it's probably not all or nothing. Or am I wrong?
No. This is super important.
I mean, if one wants to be radical, we should take everything, put it in the dustbin, and say, okay, folks, we self-deluded ourselves. We should start from scratch. But it seemed not to be entirely the case. Just give some hope to our PhD student.
Yeah. No, absolutely. So look, this is a couple of important points to make. So first of all, the results that I've showed you are all based on experiments that were not accepting of the framework that I'm advocating for. And that's important to notice because, yeah, no, those things told us something really important. All those experiments told us something really important. But when you look at them as a whole, it tells a different story. So I'm absolutely not saying that this is useless, that the scientific advances didn't happen. They absolutely did. We know an immense amount more about the brain now than we did 50 years ago. No question. But some of the fundamental assumptions under which those data were gathered turn out to be not supported by the data themselves once you aggregate them, right? And so hopefully this means we can advance beyond the assumptions that we had then. Now, as to the particular point about something like Parkinson's and the discovery that if you stimulate a particular region of the brain, you can alleviate some of the symptoms. That is not the least bit incompatible with the picture that I'm talking about, because of course what we're talking about is intervening at a particular node in a network, and then having the effects of that intervention spread through the network itself, right? So what we need to avoid is the notion that I've discovered the Parkinson's area or something like this. No, that's a silly way to think about it.
Yeah.
You've discovered a location where interventions can be especially effective. And why? Well, because of the place of that network, the place of that node in the overall network, right? If you want to disrupt or enhance Delta's flight network, where do you intervene? In Sarasota? No, you intervene in Atlanta. And why do you intervene in Atlanta? Because that's the central location for the particular structure of that network. And so I think we need to start to accustom ourselves not to giving localization, right, localist, as it were, explanations when we discover important things, say about disease, but instead to say, well, this is an especially important place where I can intervene. And why is it important? Well, that's because of where it is in the overall network structure.
So, developing your idea, you were showing the different functionality of a given single neuron in C. elegans according to different constraining contextual situation. So in a sense, once we get a much sharper technology, in principle, functional neural, we should be much better off functional neuroanatomy with non-invasive system in humans directly, where you can carefully monitor different contexts with respect to standard neuroanatomy, where you inject a given brain region in an anesthetized animal where the contextual situation is always the same. The animal is unconscious. So you cannot monitor or modulate different environmental constraints that may lead you to see that given area project also to different parts of the brain. I don't know.
Yeah. No, I think this is exactly right. I mean, obviously, advances in the field are going to depend on technological advances, but crucially, I think also on theoretical ones, in the sense of just starting to be aware of the fact that those contexts do matter, right? And that when we do experimental controls, we're not necessarily getting at the ground truth, because we may have controlled away part of the phenomena that we want to observe. And so, as you say, if we anesthetize an animal, right, the famous cats example in anesthetized cats, and we find these simple and complex cells that respond to particular. But actually, probably in a behaving animal, those cells will in fact also respond to lots of other stuff that we're not able to actually observe in that state. And so, part of the mindset change that I think has to happen here is the recognition that that we're not dealing with a system that's, say, as simple as a physics experiment. We're dealing with biological systems in which we have complex causality, and when we control for various variables, we're not uncovering the ground truth, right? We're noticing a phenomena that occurs under a circumstance, but that's not necessarily going to be the full range phenomena that particular bit of the system can exhibit.
Yeah, I think I'm totally with you and pointing out the necessity after spending your whole day in the lab, sit and think about more theoretical. I think what we have now is too many empirical data and too little theory trying to make sense in such an integrated way as you beautifully did for us today. Okay, let me ask if someone in the audience has questions coming up. Don't be shy. Okay, so if there are no further questions, we don't keep you busy any longer. I want to thank you so much for this great opportunity. I think we very seldom, or we probably never met in person. Hopefully,
We met actually at Columbia, didn't we?
yeah, but yeah, a few years
That was
ago.
a long time ago.
Yes. Okay, it was great having you with us today, and my best wishes for your future project, and I hope we'll be in touch soon again.
Thank
Bye.
you. Yeah, you've got that thing coming up in Italy. Hopefully, I can make it over there if that goes.
Thank you. Thank you so much.
Thank
Bye
you.
bye. Bye.
Bye.